How do you measure a protein? And maybe more importantly, why would you ever want to? Do you use a scale or a ruler? Well, not exactly. So let's start off with what exactly is a protein? We all know that we eat proteins. Foods like meat and eggs and cheese are full of protein, and that is why they're good for us. And bodybuilders like to eat extra protein in the form of muscle building shakes in order to help them build more muscle. Proteins are actually large molecules, well, large for molecules anyway, that are made up of long chains of smaller molecules called amino acids. So these really long chains are then folded up into super complex structures of different shapes and sizes, which we can model on computers to look like the ones that you see in front of you. So most people have probably heard of genomics and the genome, which is a study of all the DNA and genes in your body. But then there's also proteomics, which is a study of all the proteins in your body. If the DNA is the blueprint for your body, then your proteins are some of the bricks that hold you up and keep you together. They're also involved in pretty much everything that your cells do. So where DNA is made up of 4 base pairs, proteins are then made up of 20 different amino acids. And these different amino acids, organized in different orders, can code for every protein in your body. There are thousands of different types of proteins in a single cell, and if we want to see what's happening in that cell under different conditions, we can look at how those proteins are changing. For example, we can look at what proteins there are and how much there are in both healthy and diseased mice to get a better idea of what is happening when we get that disease. So one way to identify these changing proteins and to measure those changes is mass spectrometry-based proteomics. So a mass spectrometer is a really big box, and that shelf in the middle of the picture there is shown as about hip height. And this big box measures how much molecules weigh. So when you start out a proteomics project, you start with a sample. So you could start with a set of cells, or a particular mouse, or even some human samples. So then, once you have those samples, you need to take the proteins out of them. And we have the chemistry to do that. And then you chop them up, usually using an enzyme, which is another type of protein, called trypsin. And that cuts them up into smaller pieces of protein that are called peptides. So the peptides then get sprayed into a mass spectrometer. And the mass spectrometer weighs out all of the individual peptides. So inside the mass spectrometer, each of those peptides then gets broken down even further into smaller ions that are called B ions and Y ions. And those are just the original peptide with more and more of each end broken off of the peptide. So all of these ions are then measured, and that gives us something we call a spectrum, where each of the lines shown represent a certain weight that corresponds to each of the B and Y ions. And so by looking at the spectrum, we can identify what that peptide is. So one mass spec run will give you thousands of spectra representing all of the different peptides that were in your original sample. You could spend years going through that data trying to figure out which each of those spectra represented. Or you could use a computer. So computers can calculate what peptides are in your spectra based on the known masses of all of the 20 amino acids. So then we also have databases full of all of those known proteins in humans and mice and a few other animals that can be used, and the computers compare all of your spectra to those databases. So the computers first help us identify the peptides from our spectra and then turn those peptides back into proteins. But then we just end up with this giant list of proteins. And based on how often we see each one, we could tell, relatively, how much of it is in our different samples. But then we still just have this big giant list that's full of a whole lot of information. So one of the best ways to tell which proteins on the list are important and which ones aren't is to talk to a biologist, and that might be yourself or it might be someone else. And you can talk to them about which proteins make sense in the particular system that you're looking at. So then you can follow up with those proteins with more experiments. So how do you measure a protein? You start with an interesting biological sample, and then you use mass spectrometry and computers and databases, and then you interpret the proteins you find to see what's happening biologically.